It is my great pleasure to introduce the next speaker and the next session, which, of course, as I'm chairing, must be on ligands. I do have to admit that I forgot that we'd called this session Ligands Everywhere All at Once when I named my talk Everything Everywhere All at Once, but please forgive me for that. It's a pleasure to introduce Judit, who has been at CCP4 dev meetings since I started going to them and was always the industrial voice in the room. And so it's a pleasure to introduce her and to hear about how stuff is actually done in the real world in industry. So please take it away.
Thank you, Nick, for inviting me and also for almost revealing my age. When I was asked to give this talk, I thought, well, I don't know how I can contribute other than just telling everyone what it's like in industry. So I went around and asked my colleagues what they thought this talk should be about. So in this talk, this is what I'm going to start with, just telling everyone what my colleagues and other people who I talk to thought this talk should be about. Then talk about how industry might be different from academic research, and then how automation information management can contribute, what the decision-making points are, and just finally round up with a couple of thoughts. This should be more thought-provoking than prescriptive, I hasten to add. So when I asked people, they came up with a couple of interesting answers. One was, it's a little bit like, should I stay or should I go? For the very young people in the audience, this is a song that was written about 50 years ago. Go and Google it. It might be on Spotify still. A lot of people said things like, how do we know when to stop? How do we know what software to use, what hardware to use? Is my answer to the question fit for purpose? Am I answering the right questions? There was a really strong voice emphasizing early attrition. That's a big thing in industry. And there was one that I really, really liked, and that was, well, have you not read the Kierkegaard Either Or? If you haven't, go and read it. It's amusing, the first part. The second part is slightly heavier, but an incredibly important sort of philosophical work in decision-making. It says, I'll read it out briefly, my honest opinion and my friend advice is this: either you choose to do something or not to do it, you will regret both. This is the first part of the book, which is written from the perspective of a hedonistic person in science that is doing science for the sake of science, for fun. And whatever you do, you're choosing to do the opposite of the alternative. And that is where the element of regret and remorse comes from. The second part of the book says something else that is written from the perspective of more of an ethical person. Or religious, whichever way you want to look at it, where there is a big picture that is followed or adhered to, where decision-making is a little bit easier, and there is a little bit of less element of remorse or regret in the end. So I'll come back to that later. Why do we need to make decisions in industry? Well, or in anywhere in structural biology. In an ideal world where we're riding unicorns through a fairy meadow, we have unlimited time to do everything, unlimited manpower, machine power to do absolutely everything, and we work with optimal reagents and tools. In real life, that rarely happens. We are riding camels through deserts with a fraction of a person's time, limited machine computing time. Everything is just literally totally suboptimal. So we need to make decisions to get the best out of our limited resources. In industry, that's particularly poignant. This is a very business-like chart, US-based, showing the number of new drugs, that's a pale blue, versus the R&D expenditure in the pharma industry, big pharma. And what we can see is that there is a disconnect between the output and how much resources pumped into this process. So there is a pressure based on this to do what we do faster, cheaper, and smarter, making better decisions. Also, I don't know if you know, if you don't, here's a little tidbit of information in the US. Now, the time point at which a drug's price at which it can be sold is renegotiated at an earlier point from its launch. So particularly for the small molecule drugs, there is a big pressure to produce them cheaper and faster. And that trickles down to structural biology as well. Going back there. So the process that we do is exactly the same what an academic researcher would do: design constructs, produce the protein, get crystals, put them in the beam, process data, solve the structure, build a model, validate it, end of story, right? This is a series of decision points. Each step informs the next, or the previous one if the result is negative. I hasten to add that this is not a linear process, it's iterative. We tend to go back to the previous one quite a lot. Lots of it can be automated, as we heard and will hear, which is not necessarily the best thing to do, but can help. But the decisions made always based on incomplete data. I'm not talking about crystallographic data here, well, that too, but so like the scientific background based on which we need to make our decisions, which is, if you're a scientist, it's really uncomfortable. If you work in management, that's a different kind of story. It's also a numbers game. So ideally, I'm not going to read out the numbers, but if you multiply all of these in an ideal world situation with the unicorns, there could be millions of individual experiments performed to get to a structure. Now, that rarely happens. We rather have maybe time and resource for 15 constructs per project to look through enough protein to set up 5 plates. People have been there, I guess. Only an hour on a beamline, and the chemists are already breathing down my neck to share the results. The first strategy, trying to pretend that we live in a perfect world, is a valid one. That's brute force. That is basically why we have all the wonderful pipelines and technology to help us with all the automation. And this is basically where all the genomics projects succeeded, or how they succeeded 15, 20 years ago. And that is what industrial crystallography maybe was based on at some point. In the real world, of course, we need to make decisions, as we said, but still make good use of automation, etc. I'd like to go back to the process and what I said about this whole philosophy thing. And that is that if you don't put this process into the big picture, which is our aim, and that is to deliver life-changing medicines to patients, the processes and the decisions that we're making is fairly meaningless. So everything will be driven by the big picture, first of all by the study design, which would ask a specific question, set a period of time that is needed or should be used to get there, and define the resources. This will result in easier decision-making, as I said, and help us decide whether we are answering the question, the right question. Is our result fit for purpose? Is it feasible in the first place? And of course, hopefully, we will regret our decisions a little bit less in the process. It isn't only the sort of starting point of the project that is important, it's also what happens beyond structure deposition, whether that's internal or external. One could imagine that driving drug discovery, sort of top right corner, will require different kinds of experiments and results than saying epitope mapping or basic research or mechanistic studies. Those are completely different kettles of fish, and we'll need different kinds of project plans and decisions to be made. You can get away with mediocre resolution for epitope mapping. For drug discovery, you need a little bit more, and you can't see hydrogens unless you go to atomic or subatomic resolution, of course. So that makes that kind of decision-making fairly easy. So industrial structural biology has multiple different ways it can impact the process being first selecting targets, generating lead molecules against that target in a small molecule world, optimizing those to clinical candidates, and then pushing those through phase 1, 2, 3 clinical trials, finally launching the product. Structural biology impacts mostly at the research part of this. That's the mulberry purple part. The gold is what is the development of R&D. Well, we have less impact there, still some. So what do we do? We can solve novel structures that are really useful for all the in silico methods. We can do X-ray screening, so basically using crystallography as a primary screen. You can do structure-based drug design. that can continue up to the clinical stage of the project, or even beyond when we're working on backup series. Slightly different questions are answered. We also have the odd request when the chemist comes up to us and says, what's in the tube? What's the chirality? We always say crystallography, protein crystallography is a really expensive way to answer the question, but questions are coming. All of these feed into what we call iterative crystallography, where we are solving the same kind of or similar kind of structures, same protein with different ligands over and over again. So what I will say in the next couple of slides will focus mainly on that. Might be useful elsewhere as well. We make use of automation and information management quite a lot. Reasons for using automation is basically cost. For repetitive, highly parallel tasks that do not require too much of brain power, or brain power that can be replaced by machines, or miniaturization, making your protein last a bit longer. It can be more objective than people making those decisions, and information handling is surprisingly easier if you leave that to machines. Or all of the above. So for that, we have the wonderful pipelines that are quite popular across industry, Dimple, PipeDream, etc. Or just automating the whole processing is in case of smaller biotechs that do not have an established structural biology group. Of course, there are drawbacks, which is that there are less bespoke experiments that one can do in a pipeline. And as a community, we're losing expertise because of that. On the information management front, there are reasons and benefits, loads of them. First of all, searchability in a multi-user environment. We work as a large group sharing projects, and if someone goes on holiday, the project needs to go on, so we need to share our data. We're linking with robotics, starting with the protein scientists feeding through to generating crystals, imaging them, sending the information, well, somewhat blinded to the synchrotron, getting data back. It's all involved in one system, and this is something that I would like to write up in the proceedings. Data provenance and reproducibility is key, and there is actually a legal requirement to capture what we did and when we did it. There are lots of platforms to do that, databases. It's been talked about in the last couple of days, workflows and pipelines, electronic log notebooks. These are really powerful when they're integrated across the whole organization and throughout the whole project. Of course, no one likes to input data, but isn't it wonderful when we can finally search and find what we're looking for? So there isn't always kind of like a balance between the two that we need to consider. So going back to study design, it should be very clear and specific on the scope, the resourcing, and the impact of what we're going to deliver, as specific as it can be. Now, there is always, a plan is great, but it's only valid until you lift the pipette for the first time, because the reagents are not going to behave the way you plan them to behave. So there needs to be a plan B in each and every step, which is not project creep, and executed before we make a decision. Negative results are results too. I can't emphasize it enough. If something doesn't work, it's not a bad thing. It means that we failed at a certain stage and we stop that process or stop that activity. We can free up resources to do something that actually works. That also can inform the next step or the previous, or just completely inform the next project design, like completely change gears and change tracks and go to different methodology. How can we get this right? Well, we look at feasibility, we look at the literature, the competitive landscape. Whatever is out there, however, needs to be taken with a large pinch of salt, maybe a bucket full of salt. In this example, one of my colleagues has been asked to scope a project based on some really exciting literature results. A binding mode for some known binders was finally solved for this protein class, and everyone got really excited about it. Until my colleague looked at the actual data and found that, well, the compound isn't really there. At which point, the whole project was nipped in the bud, and we didn't waste time and resource on something that was actually not feasible. The paper and the accompanying PDB deposition has since been retracted. Construct design, again, we're reading the literature and designing our constructs based on that, AlphaFold, etc., make this process extremely easy, also very visual to communicate with protein scientists. I'm not going to read out all the different aspects that lead to the final construct designs, but let's just say that this is really the time for brute force, because construct design is cheap, but the price that we pay working on the wrong construct is actually immense. So we can cast the net wide and see what we find. This is an oldish example that I thought I would revisit here, which is an old kinase domain that we just couldn't get the structure of. It just wouldn't crystallize. The three other examples or isomorphs in the family, they did. And the question was, why is that? Where is that single point mutation that would make this protein crystallizable? So after a lot of construct designing, which means loop deletions and surface intermediate mutations, etc., there was this one mutation that improved thermostability and led to a 2-point-something angstrom resolution structure. Now, revisiting this a couple of years later, using the AlphaFold model, it's the same region where the mutation in question sits, we can see that actually even the AlphaFold prediction couldn't have really helped with this particular design. It would have suggested that that part of the protein just wanted to be disordered and removed from the core of the protein, which actually in real life didn't work. So that means that even going against something like the rational design can prove helpful later. Going to protein production, cloning isn't really the bottleneck for us. Outsourcing and automation helps immensely, keeping expression hosts downstream in mind. What really helps is expressing and purifying the protein small scale with brute force, with automation first, and then assessing which winners to take forward is that the thing that really helps. The really good protein reagent is the main bottleneck. So we can talk about how to be more clever in crystallography until the cows come home. But if we don't get our protein reagents right, the process is going to fail. Basically, in this case, it's worth considering all possibilities and push them through on a small scale with brute force. Continuing with crystallization. Now, this is where we utilize most of the automation, information management. Basically, the bottleneck is still the amount of quality of the protein, and there is a diminishing returns on how many different crystallization trials one can set up. There is a huge scope for those wacky experiments where you would just add some trypsin that you find in the cupboard, or lace your protein with a bit of brandy from last night, which automation doesn't really give a good platform to. But if it works, that's huge. What is really emerging these days is AI and machine learning based image annotation and crystal detection. It's not 100% trustworthy, but it saves time, and time, well, time is money and an important resource. So what I would like to add, sort of industry specific, is basically the requirements for iterative crystallography, which is that it needs to be reproducible, it needs to be at the right resolution that is fit for purpose, it needs to be compatible with ligand soaking, so DMSO tolerance, maybe echo dispense friendly. Just a word of warning, low viscosity, for instance, MPD, containing plates do not fare well in an echo. And it needs to be in a physiological pH. So meeting these criteria will save a lot of time and resource downstream. Data collection. Now, I would like all the global phasing people to please cover their ears for the next couple of minutes. We mount the crystals manually. Still, there are a lot of platforms out there, not a lot. There are some emerging platforms out there that help with automated crystal mounting, but it's still in our world is largely manual. Data collection, however, is mostly automated, except for a couple of cases where it is hard to communicate expectations of prior knowledge or the experiment hierarchy. And basically here, the design of the whole experiment and the goal of the experiment is what gives us help or helps us to make the decisions. And there are, of course, all the alternatives that has been talked about recently. So here's the unpopular opinion. For us, brute force data collection is often sufficient for standard cases. Point, shoot, ask questions later, 180 degrees at a random starting point. Simply because strategy calculation, it's still taking longer than actually just getting a data set and asking all the questions later. Data processing. Now, here is the thing where we hardly ever do anything manually. It's all automated. These days, synchrotrons are wonderful at providing on the fly and beyond data processing that can be geared towards the target in question. It is really, really hard to beat manually. There are, of course, those pathological cases when we need to do something about it. But really, this is the case where automation just shines. The only question that we ask ourselves is which one of the many data sets that we get for a particular experiment, i.e. protein plus ligand, which one is the data set that we should be taking forward for structure solution, or is it all of them? Here is an example. Well, all of these look a little bit pathological in many ways. And you can pick your favorites. If you know what the true space group is, that gives you an indication, and also how and what for the data set is going to be used in downstream processes. My favorite is towards the bottom, by the way. Phasing and refinement happens in the same kind of workflow in our world. It's rarely the bottleneck, because since AlphaFold we solve everything by molecular replacement, let's face it. The decisions that are made, or the questions I often get is, what software or pipeline shall I use for the best results? And the answer is often, whichever the one is that you're most familiar with and fastest at, because that's going to be the best. Search models, those can be tweaked. Which one is the relevant confirmation? Are we using domain segmenting, etc.? The best solution is determined by which one is behaving best in refinement. There could be fine-tuning of parameters, but it's rarely needed these days. There is one thing where we make a hard decision, which is this data set worth taking forward at all? Is the ligand bound? Is it not? Is it worth pushing it further? Is it worth going back and redoing the crystallization part of it, for instance? For low occupancy binders, we've just heard the talk, there are separate workflows, and we do use them occasionally. Unpopular opinion again, so the purists, please cover your ears. Chemists rarely care about R factors. Geometry outliers, they're really pesky, but if they are far removed from the ligand binding site, they rarely influence the usability of the model. Now, this is no license for anyone to say that, well, that old bit at the CCP4 study weekend said that I can get away with high R factors and a lot of Ramachandran reactors, because that's not what I'm saying. But if you're only interested in the binding mode and nothing further, it isn't going to be published in a peer-reviewed journal. Those are irrelevant. Finally, modeling, validation, and analysis. Now, these happen at the same time, and thanks to Coot in a large extent for that. We don't do extra step of validation. It's part of the modeling process. There is a lot of automation in there, like ligand fitting, for instance, and it saves time and is objective. But as someone used to say a while ago, at least one person should have a look at the map. Again, it's a published example of how not to model something automatically. The goal is here to generate a model that answers the questions: what is the binding mode? Is the fragment bound? Where is the epitope, etc.? And also agrees with experimental data and prior knowledge, as we saw in the very first example. The goal is not to generate a model that is absolutely perfect and has absolutely no outliers. Even for publication, no model is perfect, and outliers can be real and very much valid. And aiming for perfection and spending a lot of time trying to squeeze that last little bit of refinement in is a waste of time and resource. Honesty is the key, however. So if there is something really fishy, with a model, we need to annotate that. We need to disclose it and be open about it. Because as I said at the beginning, there's nothing worse and more damaging in industry than trying to work on something that isn't there and is not working. Again, model building and validation never ends. We can revisit all data sets, and it's happening all the time. We only stop where we feel that the model answers the right question. So with that, 25 minutes, I'd like to summarize. Going back to our first sort of philosophical question of how do we make best decisions without regretting it later, study design and the big picture, the passion, the drive to get medicines out there to patients is what dictates most of the decisions. They might be scientific, they might be portfolio-based. Study plan can change. The process is not linear at all, ever. Perfection is not the aim, fit for purpose is. Negative results are extremely, extremely important for early attrition and stopping things that don't lead anywhere. There is a huge amount of importance to the data automation. It is only powerful if data is managed alongside with it. And with that, I'd like to close and happy to answer any questions.
Thank you for keeping absolutely perfectly to time. Are there any questions in the room? Please do remember to introduce yourself before asking your questions.
Dave.
Rasmus Fogh, Global Phasing, and I forgot to cover my ears. So I want to say that I think this is a really good talk, and you made a very good point that you need to spend the resources to answer the questions. I will make a small comment, which is that the strategy calculation, it does not double the time to run an experiment. It may take as much as acquiring 180 degrees, but by the time you mounted the sample, etc., that is actually a fairly small fraction. So it is definitely a trade-off, and it is by all means not sure that it's worth it, but it is not quite as bad as I think you said.
That is a very fair comment.
Dave Brown, Servier. Thanks for a nice talk. Obviously experienced a number of these problems myself. Would you like to just comment on the role of the industrial crystallographer, both in terms of two things? You didn't really mention timeliness of a structure, but also the education role that you play with your colleagues.
Let's start with timeliness, because that's a little bit easier and less controversial. In the lifetime of a project, and I'll go back a couple of slides, if I may. Oops, not that. That. Right, so in the lifetime of a project, if you don't manage to get an iterative system up and running, say, be able to accept requests for structures on a regular basis with a good success rate, by the time compounds come in from, say, high throughput screening or biophysical screening methods, the boat has sailed, and none of what we did up to that point is meaningful. If our goal is to influence chemical design, and this is what structure-based drug design is for, structure-activity relationship is all about, if we can't influence that, then all of our clever work has been meaningless. So that's the time element. And the second question was education. Very good question. I think it's fair to say that our group has spent 20 plus years educating chemists to read structures. And I think now we reached a point where there is a really good critical mass of chemists who are really proficient in reading structures. Reading structure meaning they know what electron density is about. They know how to read difference maps. They understand alternate conformations, and they know the limitations of restraining small molecule structures or substructures. They know very well what atoms we do see and what we don't, hydrogens, that all electron density is blue, no matter the atom type. So these are the things that we had to repeatedly explain over and over again. I think within the industry, chemists are very well educated now, with the unfortunate exceptions. Would you agree with that, Dave?
I think in different environments, yes.
Okay.
But I think in terms of what we talked about, I think certain chemists, and use of modeling and alpha fold structures instead of structural biology. It's not just the chemists that need educating within the organizations, it's the wider organization itself.
Again, fair comment.
Question from the Slack, I think.
One from Albert. In my experience, a simple strategy can be achieved in less than 7 seconds. More a comment, I guess, than a question.
Good for you.
Okay, one final question.
Hi, Hans Valsgraf from Diamond Light Source. Thanks a lot for a very useful talk. You showed a graph of R&D spending and approval of drugs, and there seems to be an increase in recent years. Do you think the increase in approval correlates to the increased spending recently, or we're getting good at this, or does it correlate to the increase in spending 10 years earlier because there's a lag in the pipeline?
So the article that came with the graph said a couple of things about it. First of all, there is a certain amount of disconnect. Also, there is a huge amount of variability, which is project-based, therapy area-based, etc., for how much an actual project or an end product costs. And I can only echo that is the case. To answer the question specifically, the first part of this chart takes years. So that's just the purple part of it until it goes to the clinic. And the clinical trials, again, at least that long, if not longer. That is where the cost is the most. What I think the chart reflected was the increased cost in the second part of the process, where you have to save money is basically across the board, or time.
Okay, thank you.
Okay, thank you very much, Judith. So while the next speaker is setting up,